Hello, everyone. Thank you for attending today's webinar, Population Scale Circulating Biomarker Discovery, Aligning the Right Patient, Disease, and Therapy to Accelerate Drug Development, presented by Sapient. I'm Andrea Anderson, and I'll be moderating this webinar. Our speaker today is Mo Jain, founder and CEO at Sapient. You can read his full bio on the left side of your window by selecting the Speakers tab. Just a few technical notes before we begin. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. This webinar is being recorded and will be available on demand within 24 hours after the event. Time permitting, we will follow the presentations with a Q&A session. Please submit your questions using the Questions and Answers tab on the left side of your screen. All right, let's begin. Mo, please go ahead.
Thank you so much, Andrea. Really a pleasure to be here today and share our ongoing work with you on population scale circulating biomarker discovery. As mentioned, my name is Mo Jain. I have the pleasure of representing the Sapient team today. And we're really excited to share with you some of our ongoing approaches and work and results. So the real fundamental problem that we are tackling here at Sapient is the one that many of our audiences are dealing with on a day-to-day basis. And this is the challenges with making drugs and developing drugs. And as we know, over the last decade, the failure rate associated with drug development continues to be really quite a hurdle, estimated to be greater than 90%. And we think a lot about why is that in fact the case, despite all the technologies that have emerged and all these sort of new ways of developing drugs. And one of the real reasons in our mind has to do with this fundamental mismatch between how we diagnose disease and how we actually treat disease. And so diseases are diagnosed typically by a singular end pathology, as you can see at the top of the slide here. Now, everyone who shares this pathology shares a given diagnosis, even though there may be many different approaches or pathways by which an individual may go from a normal to an abnormal state. Now, everyone who shares the same pathology is treated with the same drug, as you see here. And given the fact that there's many pathways by which an individual may develop a given disease, what ultimately results is that some portion of individuals will have a benefit, some portion of individuals will actually have an adverse event in response to the therapy, and a large number of individuals will actually have no underlying benefit. This, of course, is one of the main contributing factors in our mind, particularly in the later stage clinical trials, for why drug development fails. Now, there's a lot of evidence in the clinical literature to suggest this is in fact the case. And so if we look at over 30 chemotherapeutic agents that have been approved by the FDA, now, again, these are FDA-approved therapies that are being implemented and used today as part of cancer therapeutics. We find there's a great heterogeneity in the underlying response rate, as you can see here from this graph on the right. Now, this varies all the way from about 80% response rates to 20% response rate. And if we look at the median response rate across all these different chemotherapeutic agents targeting many different underlying targets, what you see is that the median response rate is about 40%, which means for any of these FDA-approved chemotherapeutics, over half the individuals receiving this therapy don't actually respond to the underlying therapeutic. Now, this is where biomarkers emerge as so critical. Biomarker-guided drug development allow us to begin to align individual patients with their specific disease pathobiology and ultimately the right therapy. So if you remember the slide we showed you earlier whereby everyone receives the same therapy and only a portion of individuals respond, what biomarkers allow us to do is to take a population of individuals, all of whom share a singular diagnosis, and isolate them out into groups according to different pathways by which they may develop a disease. In this way here, you can see that there's 4 different groups of individuals, all of which are identified by a different circulating biomarker. Now, for each of these different populations of individuals, they can be treated with a different therapeutic that's specifically guided towards their mechanism of disease. And in doing so, you can greatly enhance benefit, essentially enriching for those individuals who are going to be responders versus non-responders. Now, there's quite a bit of evidence, once again, in the clinical literature to suggest that this is not a theoretical sort of success, but is an actual success. So this is an analysis by Andrew Lowe and his group at MIT. that details over 20,000 clinical trials that have been performed over the last several decades across all therapeutic areas. And what you find is that there's one singular feature that defines those assets that have a higher rate of FDA approval. And that is those drugs that are developed together with an underlying biomarker. This is the same for small molecules, large molecules, gene cell therapies, et cetera. So it's independent of the underlying modality and independent of the underlying therapeutic area. So you can see here on the slide for oncologic therapies, those developed with a biomarker relative to not have almost a 7-fold increase in underlying FDA approval. And similarly for cardiovascular disease, autoimmune disease, infectious disease, vaccination, et cetera, et cetera. If we look at all clinical trials across all therapeutic areas and modalities, once again, you see the cumulative improvement in FDA approval is over 2x, as well as the time to approval is over 20% faster. You can imagine how this can be transformative for a drug development program to have such an increase in approval rates as well as time to development. In addition, biomarkers allow us to develop smaller, much more targeted and effective clinical trials. So this was an analysis performed by the FDA in one of their white papers on biomarker utilization as part of clinical studies. And essentially what they're showing here is as you're able to enrich a population within a given particular disease through the use of biomarkers, you can drive a much smaller clinical study. So a clinical trial that has a biomarker that isolates 100% of patients relative to 25% of patients, essentially this clinical trial can be about 20-fold smaller, which will result in not only a much more efficient trial, a higher efficacy rate, but a much quicker and cheaper sort of end product. Now, again, this is not theoretical, but has actually been borne out in the clinical study literature. So if we look at trastuzumab for breast cancer, as many of you are aware, initially this was tested in a non-enriched clinical trial across thousands of individuals, as shown on the left. And ultimately, this clinical trial, given underlying heterogeneity, as we described before, was negative in that there was no significant difference between sort of populations. Once a biomarker had been developed, in this case, the HER2/neu immunochemistry for this particular therapeutic, a subsequent clinical trial, which we call enriched here on the left, was 16-fold smaller. And as you see on the right now, in this enriched clinical trial, showed a difference in ultimate outcomes that led to the ultimate approval of this therapeutic through the FDA. So there's quite a bit of actual evidence from the real world literature to suggest that this approach for biomarkers can be incredibly efficacious in driving drug development programs. Now, to date, the majority of biomarkers that have been utilized in the clinical literature as part of these clinical studies have been genomic markers. And one of the real challenges is that when we look across human disease, the risk for most human diseases is only partially encoded in the genome, and is actually the majority of population attributable risk is not related to the genome or is non-genetic in origin. Again, this can be studied and modeled in large populations of twins using both mono and dizygotic twins. Monozygotic twins, of course, sharing complete genetic material, dizygotic twins sharing a portion of the genetic material. And when you look at the development of disease, you can calculate the proportion of risk that comes from genetic factors or the heritability of each of these common diseases. And again, when this is done across tens to hundreds of thousands of twin pairs, what you find is for the vast majority of age-relating diseases, whether it be cancer, heart disease, neurologic disease, lung disease, GI illness, NASH, et cetera, only a small percentage of risk, somewhere in the order of about 20%, is related to your underlying genome. That means 80% of risk is non-genetic and has to do with the exposures that we experienced over the course of our life. Everything we eat, drink, smell, smoke, how one organ system communicates with another organ system, how our microbiome works, how our diet works, et cetera, And all of these critical non-genetic factors are key drivers of underlying disease and are key areas for which we need to develop biomarkers. This is a white paper put together by McKinsey and Company in understanding where genomics is going to go in the field of biomarkers. And I believe this quote really perfectly summarizes this idea. And so I'll read it for you here. While genetics is a powerful indicator of disease and reflects our individual variation, it does not necessarily inform the current biological state, which is a most predictor indicator of health. Current functionality and by extension dysfunctionality, is better reflected by the transcriptome, proteome, and the metabolome, which we like to think of as small molecule metabolites or small molecule biomarkers. And this is where Sapient has really focused much of our attention in this area of what we call small molecule biomarkers. These are markers that are typically below 2,000 Dalton in size, of which there's tens to thousands of these circulating in our underlying blood. Now, these factors read out both genetic and non-genetic factors that contribute to human disease. These are molecules that are made both in normal tissue, in diseased tissue, as well as in the tissue microenvironment. And given their physical chemical properties, these can be readily released from the intracellular space into the circulation, where these can be captured non-invasively to reflect what exactly is happening at the level of the tissue or at the level of certain cells. Now, these molecules are among the most ancient in mammalian biology and are highly conserved, which allows us to cross-translate from preclinical models to human studies, which is also quite efficient as a means for discovering these small molecule markers. Now, we oftentimes think about these small molecule biomarkers that are shown here on the right in relation to genomic and proteomic markers. And while genomics have been transformative for understanding the genetic origins of human disease, these really represent a static measure that are set for the most part from the time of birth. Protein markers can inform on some acquired biological processes, but once again, only a small portion of the underlying proteome is released from the intracellular state into the blood. Just to reiterate this point, small molecule biomarkers, in contrast, are highly dynamic markers that reflect the underlying state of the individual as well as future diseases and capture not only genetic factors and static measures, but also dynamic processes related to lifestyle, intra-organ communication, the microbiome, exposures, diet, and many other factors which are absolutely essential to the development of disease and ultimately drug response. Now, the challenge to date has really been around how do we go about measuring these small molecule biomarkers? This is not a new idea that these can be incredibly transformative for clinical medicine and drug implementation, but the measurement of these has really been challenging. And this is where Sapient has made great strides in the generation of what we call next generation mass spectrometry. Now, these tools, and this is an actual picture of the laboratory that's pictured right behind me in physical space. Now, these tools allow us to take a biological sample, whether it be blood, plasma, serum, a dried blood spot or a fingerstick spot, urine, CSF, vitreous eye fluid, tears, any tissue, tumors, or cells even in a dish. And we can very rapidly measure over 11,000 small molecule biomarkers. Now, for each of these small molecule biomarkers, for those of you that are technically sort of aware of these mass spectrometry technologies, we derive what's called 4D chemical classification, measuring not only the retention time, but also the MS1, MS2 spectra, as well as what's considered the IMS or the CCS values for each of these molecules. Now, these four-dimensional chemical characterization allows us to identify 500 to 1,000 known molecules from this 11 to 15,000 measurements, as well as classify each one of these unknown molecules in a way that allows us to map them back to an underlying database that I'll talk about in a moment, as well as isolate these molecules for ultimate identification. Now, given the rapidity and the rate at which we can operate here using our next generation mass spectrometry systems, the analytical cycle time for analyzing a particular biological sample is under a minute. It's just around 52 seconds to date. And given the capacity and the automation with which we've built in our underlying pipelines, we can handle tens of thousands of samples at real scale across a given time period. Now, shown here is an extracted ion chromatogram from one of these rapid liquid chromatography mass spectrometry or RLC systems. This is a plasma sample. What you'll see that this is not flow injection or direct injection. This is not inline solid phase extraction, but it's true chromatographic separation that's occurring over a very, very rapid period. Once again, what this allows us to do is to separate very chemically distinct molecules from polar molecules to polar lipids and nonpolar lipids, all in this singular run to characterize and assay the levels of these molecules from 11 to 15,000 small molecule measures, as well as given the way in which we prepare and handle samples, reduce underlying suppression artifacts that may influence underlying measurements. Now, these technologies, as you can imagine, can generate massive amounts of data. And really utilizing this data requires integration within an underlying platform that allows us to go from data to underlying actionable insight. Now, the way we do this is integrating these technologies and the data that emerge from them with two other areas. The first is shown here in our biocomputational integration, and what this consists of is an expert data science team here at Sapient that has deep expertise in the handling of mass spectral data, the analysis of this data, and integration with other types of data sets, whether it be clinical outcomes data, biological processes, other measurements, other biomarkers, genomics, the microbiome, as well as many other circulating factors. Now, all of this information is actually encoded in what we call our human biology database. This is a database that's been established here at Sapient across hundreds of thousands of individuals in which we've already made these mass spectrometry measurements. Now, this database allows us to amplify and take singular discoveries and cross-validate them very rapidly, as I'll show you in the subsequent slide. Now, this provides an overview of our underlying human biological database. Again, this consists of hundreds of thousands of individuals, now approaching over 10 million phenotypic data points across many, many different diseases, from cancer to cardiometabolic disease, maternal health, autoimmune disease, neurodegenerative diseases, kidney and renal diseases, lung disease, as well as GI illness and other lifestyle measures. For each of these, we not only have human disease measures that are encoded in this database, but as well as preclinical data sets and preclinical models. We have measurements of other biomarkers, human biology, clinical outcomes over time, genomic, proteomic, as well as many other omic-based technology measurements. And again, this database allows us to take any discovery that may emerge from a given experiment and amplify that discovery across 100,000 people in both space and time very rapidly. Now, all of these approaches, whether it be our human biology database, our technologies, our underlying biocomputational integration, are all embedded in what we call our discovery pipeline. And this pipeline is particularly optimized to allow us to go from beginning to an underlying discovery in record time. This consists of everything from automated sample excisioning and LIMS integration and automatic sample preparation through our underlying RLCMS technologies that are able to generate data. We use supercomputing-based batch extraction and normalization to handle and extract data. Using 4D chemical classification as mentioned, we can identify very rapidly 500 to 1,000 known molecules, as well as over 10,000 unknown molecules. We typically perform very deep biostatistical analysis, including using regression statistics, as well as much more advanced machine learning and AI approaches to identify singular molecules that provide great actionable insight. We technically confirm these using traditional canonical long-gradient LCMS approaches, as well as cross-validate these discoveries across over 100,000 individuals using our human biological database. And ultimately, we can assist in the development of high-complexity CLIA assays that allow us to rapidly translate these discoveries for our partners into clinical practice as part of clinical trials, as part of drug development implementation programs, and ultimately as for the care of patients. Now, to give you an idea of what this looks like, this is an initial analysis performed here at Sapient across 50,000 individuals that are diffuse adults from around the world, to give you an idea of the degree of diversity in which we can detect using these biomarkers. If you look here to the left, what you find is across this population of 50,000 very diverse individuals, there's over 100,000 total circulating factors that are assayed. Now, among this great diversity that scales even genomic diversity, what you find is that there's somewhere in the order of 10 to 12,000 common molecules, meaning these are present in most humans independent of where they live and the way in which they live their lives. In addition, there's 3,000 to 4,000 what we call universal features in individuals and humans that are required for us to walk and talk. And this includes common sugars, common lipids, common nucleotides, for which all of us have these present in our blood. Now, as mentioned earlier, these small molecules can be quite dynamic, and it's very important that we understand the dynamic nature of them in order to leverage their underlying biology for drug development. And so when we take these molecules and we look across a 24-hour period, as expected, many of these molecules change markedly. So over 50% of these molecules change more than 50% over the course of a day in relation to eating versus fasting, sleeping versus being awake, exercise, walking, circadian cycles, etc., all of which can influence these molecules over the course of a day. Now, quite surprisingly, that if we look in first and morning fasted blood samples and come back even several years later, you find that these molecules are actually remarkably stable at a given time and day in individuals. And this has to do with our continuity of exposure and the way our organs work. And so these molecules change very little over a period of time when we're looking at a first and morning fasted sample. And this is what allows us to use this information as part of these drug development programs, as I'll show you in just a moment. Now, across twin studies, we can begin to estimate the influence on these markers from genetic factors to environmental factors and shared environmental factors. And when we do these triumvirate analyses, what we find is that the vast majority of these molecules are partially controlled by the genome, but the vast majority of influence comes from environmental factors, which are once again critical for capturing this other 80% of information, which is not encoded in the human genome. Now, we also work quite a bit on the integration of these small molecule biomarkers with other types of data, including genomics measures. Shown here is a genome-wide association across these hundreds of thousands of circulating small molecule biomarkers in relation to millions of underlying polymorphic genetic variants in humans. And we have this information encoded in our human biology database across more than 20,000 individuals. And when you look at this data, what you find in this modified Manhattan plot is that there's hundreds of genetic variants which can influence these small molecules, and integrating this information with genomics can have enormous potential for drug discovery, as we'll just show you in a moment. Now, if we look across the entire genome, what we find is just under 50% of all the molecules we measure in blood have at least one genetic variant for which it is strongly associated at a genome-wide statistical threshold, meaning that for over half these molecules, there is some genetic component that's regulating their levels that we can interrogate and classify. Now, across over the 20 million SNPs that are part of the human genome, we find that just over 5% of these common polymorphisms have a circulating marker for which it associates. Now, if we enrich specifically for those genetic instruments that are within the transcribed gene regions, these are the polymorphisms that actually affect the protein encoding entities, what you find is that over 90% of SNPs that occur in transcribed gene regions actually influence or have an associated small molecule biomarker in blood across our underlying database. And this is across all 20,000 genes that are part of the human genome. What this allows us to do is to very quickly go between genetic variants and small molecule biomarkers and vice versa in a way that can be very informative for drug development and understanding human biology. For instance, we can use these integrative analyses between genomics and small molecule biomarkers to inform on biological mechanisms, essentially understanding how genetic variants may give rise to various disease states. In addition, we can leverage this information to provide readouts of genetic targeting and as metrics for target engagement, essentially identifying small molecule biomarkers, as we'll show you in some examples towards the end here, that may read out actual activity or drug engagement of a specific target. Ultimately, what these small molecule biomarkers or the integration of these biomarkers together with genomics allow us to do is to understand those disease pathways that are relevant for a particular disease state, and in doing so, identify new targets for potential therapeutic intervention. Now, as mentioned, these underlying small molecule biomarkers are extremely powerful readouts of genetic targeting or target engagement. And to give you a schematic view of how this can actually work is through these genetic instruments, we can identify those circulating molecules in blood that read out changes in particular protein activity, whether it be a direct target of a particular therapeutic or a secondary pharmacodynamic marker that may emerge from the activity of a particular drug. Now, this works quite well both for gene therapies, which are silencing a particular genetic target, as well as for small and large molecules that may be antagonizing a particular biological pathway or a particular protein. Once again, given the physical chemical properties of these small molecules, they're produced intracellularly and then released into the blood, where we can capture them and measure them by these technologies as a means of providing a non-invasive view to what's occurring at the level of the target tissue, which is an extremely powerful method for understanding how drugs may be working and prioritizing patients for clinical studies. Now, as a whole, what these biomarkers allow us to do is essentially they're a tool that allow us to apply and align patients, diseases, and specific therapies. As you can imagine, there's many different potential applications of these underlying approaches and these discovery means. When thinking about the right patient, they allow us to stratify patients using these small molecule biomarkers into responders versus non-responders. They allow us to perform safety profiling and understand those individuals who are at high risk for development of an adverse event from a particular therapeutic. They can be leveraged for companion diagnostics as part of a drug development and implementation program. And they allow us to begin to enrich clinical trials, once again, performing smaller, more targeted, highly efficient clinical studies that can be far cheaper, far more efficacious, and far quicker to perform. They also allow us to begin to understand specific disease characteristics, including disease mechanisms. They allow us to elucidate new targets for potential intervention. These small molecule biomarkers can be leveraged, as I'll show you in just a moment, as early disease detection markers that allow us to diagnose and detect disease at its earliest time points, as well as understand disease progression, essentially identifying those individuals who are going to have a very rapid progression relative to a much more indolent disease progression. Ultimately, they allow us to take these individuals, understand their disease processes, and then ultimately align them with the right therapeutic, including the right dosing, the timing, evaluation of target engagement, and ultimately pharmacodynamics. In totality, what this allows us to do, once again, is accelerate the underlying drug development program with an eye towards enhancing patient outcomes and developing drugs in a much faster and cheaper method. Now, Sapient very much operates as an organization that works together with our underlying clients. Whereas we're not a traditional CRO, we very much view ourselves as a discovery partner that works to accelerate discovery specifically for our clients. There are many different modes under which we can operate, everything from a service-based organization through licensing of discoveries that have emerged from our human biological database, and even co-development with certain partners. And so we'd be happy to talk with anyone who may be interested in working together to accelerate their drug development program. In the last 10 minutes or so, what I'm hoping to do is just to spend some time talking through some real-world applications. I recognize what I've shown you today is largely theoretical until you can actually see real-world data. And so we want to share with you some examples of how we've leveraged these technologies, the data that emerges, and the underlying approaches in order to develop and discover key biomarkers for disease states. The first example here is a discovery of an early diagnostic marker that allows us to diagnose individuals as well as elucidate new targets for intervention. Now, there's many different diseases in which Sapient works, as mentioned, our human or biological database, and one that we're particularly interested in is liver disease or NASH. Non-alcoholic steatohepatitis, as many of you are aware, is an extremely complex disease, incredibly prevalent, but very hard to diagnose in humans, and typically requires invasive liver biopsy, which is quite a morbid and dangerous procedure. And so we've worked together with partners to identify circulating small molecule diagnostic markers for underlying NASH. Now, the way we've approached these studies, as shown here on this slide, is to start from a population of over 10,000 individuals in which we have clinical phenotype data, and we know over time who develops a syndrome consistent with NASH by both liver enzymes as well as fat imaging of the liver. And when we take these 10,000 individuals and compare them to those individuals that do not develop NASH over time and analyze their blood, what we can do is measure these thousands of markers, capturing across these 10,000 individuals more than 50,000 circulating markers that are shown as these little gray dots on this volcano plot here to the left. Now, if we ask, are there markers that are specifically altered in individuals that go on to develop NASH versus those individuals that are controlled for age, gender, demographic features, glucose levels, etc., but do not develop NASH? What we find is that there's over a dozen molecules that are shown as these red prioritized dots on this plot here on the left that are drastically altered after accounting for multiple hypothesis testing in individuals that develop NASH. Now, as mentioned, the vast majority of molecules that we're capturing are unknown molecules, and only a small percentage of the small molecule biomarkers floating around in your blood today have ever been structurally elucidated. As is often the case, the most interesting molecules here, again, the ones that are shown in red, all represent unknown molecules that have never been assayed and never been characterized in humans or in others' data sets. Now, when we look at these markers, there are ways in which we can begin to organize these into biological pathways, and one which approach by doing this is through molecular networking. Essentially, we can take all of these unknown molecules, and by looking at their fragmentation patterns, we can cluster them according to biochemical pathways, which is what's shown here in the middle image. And when we do so, we find that 3 of these markers that are shown in blue all fall in the same biochemical pathway. These are all unique molecules, but also have very shared chemical features that allow them to work together and operate in the same biological pathway. Now, we can take one of these that's shown here as the key biomarker, isolate it out, solve its underlying structure, and develop diagnostic assays around this. We can also validate this in independent, both human and preclinical studies, as we've shown on the right here. Now, when we look at this one singular biomarker here, again, this is not a panel of markers, but just one single biomarker. When we look at it in additional human studies in individuals with biopsy-proven NASH, and so these are individuals who have undergone biopsy and have a pathologic diagnosis of NASH, what you find is with increasing stage of NASH severity, the molecule is increasing in human blood. Interestingly enough, what we find is that in those individuals that all have stage 3 NASH, so these all have a very shared pathology again, we see that there's great heterogeneity in underlying levels of this marker. And we see this on the top right panel here. There's a population of individuals with stage 3 NASH that have very, very high levels of this marker. And there's another population of individuals with stage 3 NASH. Once again, these are indistinguishable by pathology that have lower levels of this marker. Now, we have reason to believe that these individuals may have developed NASH for very different reasons and may respond to very different therapies. And this is once again important in being able to isolate out and substratify a population, all of that have a shared diagnosis, into those that may ultimately be responders versus non-responders for a particular therapeutic. Now, this is a molecule we can also validate in preclinical studies. Once again, these markers are among the most ancient of markers that are highly translated down to preclinical models. And so when we look at a preclinical model, we see that this is a marker that elevates in blood at the earliest sign of hepatic inflammation at several weeks. Whereas those individuals, or those preclinical animals that develop a precursor state of NAFLD do not have this marker in blood. We can begin to also examine this marker across greater 100,000 people now in our underlying human biological database, as I mentioned. And there's many questions we can begin to understand and amplify this underlying discovery. First, we can begin to ask, how early on in the disease process is this biomarker altered? And so when we look across 100,000 people, what we find is that this marker is elevated in individuals who ultimately go on to develop NASH up to 10 years in advance of formal clinical diagnosis. And so if we look at the plot here on the left, we find that individuals that have high levels of this marker, relative to those individuals that have low levels of this marker, even though they're matched for age, gender, BMI, demographic features, glucose levels, triglyceride levels, et cetera, we find that those individuals that have high levels of this marker have a 5-fold increase in the development of NASH over subsequent decade. As such, this is a marker that potentially may be used for isolating out these individuals for therapeutic intervention at very early stages before clinical diagnosis is typically made. In addition, we can also ask, how dynamic is this marker? How does it change over the course of a day? How does it change over the course of a year? How does it change in different geographic locations around the world and in different populations? And we find that this is remarkably stable with regards to times of day, diet, geographics, demographics. But what we do find is that those interventions that improve fatty liver infiltration actually result in a dynamic change in this marker. So if we look at those individuals before and after bariatric surgery, and as you're well aware, bariatric surgery can result in a drastic improvement in liver fat within several weeks, we find that this marker is dynamic and changes almost back down to normal states in individuals after they've undergone bariatric surgery. Now, this is extremely important in thinking about how one may leverage these markers as potentially surrogate endpoints as part of an early phase 1, phase 2 program to evaluate potential efficacy of a therapeutic at a very early time point, even before there's clinical evidence for improvement. Now, as mentioned, we can also do quite a bit of integration. Across our human biological database, we have over 20,000 individuals in which we have dense genomic measures with these same biomarkers assayed in blood. And essentially what we can do is the genome-wide association around this singular marker. And when we do so, we derive the Manhattan plot that's shown here on the left. What this allows us to do is to understand the actual biosynthetic enzymes and transport systems that allow this marker to be produced and to transport it into blood. In addition, given where these enzymes are expressed, we can actually begin to localize the production of this biomarker to the actual hepatocyte and even an organelle specifically within the hepatocyte. Now, one of the areas that we think a lot about here at Sapient is causal inference, meaning are these markers simply passive correlates of disease, which can be used as diagnostic parameters, or are these markers actually driving disease processes in a way that allow them to be actual therapeutic targets themselves? Now, there's many, many ways in which we begin to evaluate this, but in humans, we like to use an approach called causal inference through Mendelian randomization. And essentially, if we look at those genetic instruments that are shown here on the left that alter the level of this biomarker, and then ask across more than 500,000 individuals in our database, how does this actually influence the development of the disease? You obtain a graph as shown here on the right. And what we find is that those genetic variables, those common polymorphisms, again, that alter the underlying biomarker, also result in an increase in disease risk over time. And you see this linear relationship, again, as shown here on the right. And when you see this, it's extremely suggestive of an actual causal relationship between this biomarker, the pathway that gives rise to this marker, and ultimately disease. Now, this is a marker that's been elevated and advanced here at Sapient to our human clinical CLIA laboratory for development of a CLIA diagnostic test, as well as the biological pathways that give rise to this, given their causal inference, have been advanced towards new gene and cellular-based targeting mechanisms. Now, there's many other disease processes and disease areas that we work in. I was hoping to share with you some data from our rare disease program. Again, just showing you data previously in our common disease area of NASH. We also think about rare diseases, including those for which there are not 10,000 patients that are available for study. Here's a study that was performed in a rare disease genetic population of individuals that had inborn errors of the mitochondria. So these are individuals that have sequence-verified mutations that are occurring in one of the 1,000 or so different genes that affect the mitochondrion. These have clinical heterogeneity, and given the fact that there's many particular genes that may be altered and biochemical pathways that may be altered within the mitochondria, these individuals are extremely tough to diagnose and follow clinically. Now, when we look at over 100 individuals with sequence-verified mitochondrial disease and ask once again, are there markers that are altered in their blood relative to age, gender, demographic matched counterparts, what we find is the data here on the left. Again, we can capture thousands of circulating small molecule biomarkers across these diverse populations using our underlying RLC technologies. And when we ask, are there specific molecules that are altered in individuals that have mitochondrial disease, what you find here is in the inset, and that there's a subset of molecules that are altered specifically in individuals with mitochondrial disease. Now, once again, as was the case with the NASH example I just showed you, all of these markers represent unknown sort of small molecule biomarkers that have never been classified. When we look at their underlying structural relationships, we find that over a dozen of these markers, as shown here on the red dots on the left inset, all are chemically very related to one another and represent a whole new biochemical pathway that hasn't been described before in humans. Now, all of these markers are once again altered in individuals with underlying mitochondrial genetic diseases. We can isolate out one of these that's shown as a blue circle here, solve its underlying structure, develop targeted assays that allow us to technically replicate this data, as well as biologically validate these now, once again, in independent clinical sets. So when we look at an independent data set of patients with mitochondrial disease, what we find is control patients have very low levels of this marker. Patients that have very localized mitochondrial disease affecting only a single cell type within the eye, once again, have very low levels of this marker. And in individuals that have systemic mitochondrial disease, again, through genetic means, have quite an elevation in these underlying markers. Now, as was the case with NASH, even though these individuals may have shared genetic mutations that ultimately result in their mitochondrial disease, you'll see that there's a massive spread in the underlying levels of these markers. Now, this spread has incredibly important prognostic information and very much varies with the degree of disease severity that we observe in these individuals. Now we can begin to ask, using formal ROC analysis, what's the diagnostic sort of value of this single biomarker once again. And what we find is that in independent populations, the AUC is about 0.8 as a diagnostic test. This is something that beats current genetic testing in these individuals. Once again, that is quite complicated given the thousands or so genes that may be potentially mutated in these individuals. As is the case with the NASH molecule, this is a biomarker now that's been advanced to our high complexity CLIA laboratory for development of a CLIA diagnostic assay. Now, what we can do as part of this rare disease population is we can begin to ask, what are those genetic variants that associate with the circulating marker, even in normal individuals? And when we do so, you develop the Manhattan plot that's shown on the bottom right. Once again, we can localize the enzymes that produce this underlying biomarker, as well as the transport systems that allow them to be transported into the cytoplasmic space, from the cytoplasmic space into the blood. And when we do so, again, we can localize this to the mitochondria. Now, this is a marker that we've been able to validate in vitro and cellular systems, both in iPS-derived models of mitochondrial disease, as well as in fibroblasts isolated from human patients. This is also a marker now that we've validated in preclinical models across a number of transgenic animals that have mutations within the mitochondria. Now, as often the case with our rare disease populations that we study here at Sapient, we're very interested in understanding how these rare disease populations may also map onto common human diseases. Now, as you're well aware, the mitochondria has been implicated in a host of human diseases and biological processes, everything from heart disease to neurodegeneration and Parkinson's, as well as even biological aging. Now, the challenge with studying the mitochondria in vivo in humans is that there's no good measurement that allows us to interrogate mitochondrial function in vivo in humans. And this requires either isolating out tissues and studying them in very artificial systems, or measuring very indirect metrics that provide very poor insight into underlying mitochondrial function. In our human biological database, however, we can begin to ask, how does this one singular biomarker that we use as a readout for mitochondrial dysfunction, how does this map onto various diseases and biological processes, including age? And what we find is that across over 100,000 diverse individuals from around the world, there's a very, very consistent pattern whereby somewhere in adult age, around age 48, 49, this is a marker that begins to elevate in blood and linearly increases from about age 50 to approximately age 100. And this is some of the first real-world evidence in our hands to suggest that there is progressive mitochondrial dysfunction that occurs with human biological aging in vivo in individuals. Now, if we pick any particular chronologic age here, whether it be 70 or 80, and ask individuals that have higher levels of the marker indicative of greater mitochondrial dysfunction relative to individuals that have lower levels of this marker, are they prone to certain diseases? What we find, in fact, is that having an elevation at any given chronologic age, indicative of more accelerated mitochondrial aging, is very much associated with certain disease processes around cardiometabolic disease, as well as age-related tissue degeneration. Now, as mentioned, this is a marker that's been advanced to our high complexity CLIA laboratory for development of a diagnostic test that will ultimately be used in humans for rare diseases. But also, this is a marker now that may be leveraged as part of aging and longevity studies, as well as for the development of therapeutics in the cardiometabolic and other tissue degeneration spaces that are targeting the mitochondria. Now, the last example I would hope to share with you here today is around a marker of target engagement. And if you recall back to just several slides ago, one of the real areas that allow us to be able to take underlying genetics information and leverage these small molecules in order to derive underlying circulating factors that read out target engagement or genetic silencing. And this is an area that we've worked extensively in across many, many different pathways. I was hoping to share just one example with you today around the PD-1 pathway. Obviously, many of you are aware this is a pathway that has transformed oncologic therapies in the development of immunotherapies now that reawaken the immune system to target cancer cells. It's been very challenging, however, to evaluate which individuals are going to benefit from these PD-1 therapies. And we know clinically there's massive heterogeneity, once again, in responders versus non-responders. This is an area we've thought about for quite some time. And if we look at those genetic common polymorphisms, again, that occur in the PD-1 pathway, we can ask across the 50 to 100,000 circulating factors that we've assayed in our underlying database, which ones associate with polymorphic variants in the PD-1 pathway. And when we look, we find there's 4 to 5 individual markers, again, that are shown here on this bottom chart that are strongly and statistically associated with changes in the PD-1 pathway. Now we can take one of these markers, isolate it out, once again, solve its underlying structure, develop targeted assays. And when we examine this in individuals undergoing immunotherapy, what we find on here on the left is relative to pretreatment. This is a marker that's highly dynamic and increases in individuals undergoing a PD-1 or PD-1L-like therapies as part of their immunotherapy regimen. Now, even in those individuals that have an elevation in this marker, what you'll see on the graph on the left is there's quite a bit of spread in the post-treatment measurement, and that there's some individuals that have very high levels of this, indicative of greater target engagement, and individuals that have a more modest increase in this pharmacodynamic or target engagement biomarker, indicative of less target engagement. Now, if we ask those individuals that have the most robust biomarker response, how does this map onto actual response to the underlying therapy? What you find is quite startling. And that is those individuals that have greater evidence of target engagement, again, as assayed by this single biomarker, have almost a 50% increase in response to the underlying PD-1 therapy, as shown here on the right. Again, this is a marker that we can begin to interrogate in our human biological database, beginning to ask many factors and understand the biology of this. We've already begun to understand its response in immunotherapy, as well as changes across various demographics, how this maps onto other autoimmune and immune-related function, including over time, understanding how these change over the course of a 24-hour period in relation to cytokines and other inflammatory states, as well as to understand how these markers may be altered in both acute infection, vaccine response, and in relation to various immune cellularity as a means providing an understanding of how these markers may be reading out underlying immunotherapy response. I'll go ahead and stop there. I would like to thank the audience for their time and attention, and hopefully we have a couple minutes, Andrea, here to answer any questions.
Yes, we will move on to the Q&A now. There is still time to submit your questions using the Q&A tab to the left of your screen. We have a lot of great questions already, and we will try to get to as many as possible. For the first question, can you comment on the value of small molecule biomarkers relative to peptides or proteins?
Great question, Andrea. And so, as you're well aware, the vast majority of markers that have been developed in humans really represent protein biomarkers or genomic markers. And those have been instrumental, again, in understanding human disease. The value of small molecule biomarkers is several-fold. One, as mentioned, these read out not only endogenous processes that are occurring in the cells, but also exogenous processes from our microbiome, from one cell communicating with another cell, from our diet, lifestyle, as well as environmental exposures, all of which have profound influence on human health and disease over time. In addition, given the physical chemical properties of these small molecules, they can be produced in a particular cell type, whether it be a liver cell, a brain cell, a muscle cell, in a gut cell, and released quite readily into the blood, again, where we can measure these non-invasively. This is in contrast to protein markers or peptide markers, for which the vast majority of these are retained within the intracellular space and require biopsy. Now, there's a portion of protein markers that are, of course, released into the blood, including those that are related to circulating immune cells, but the vast majority of proteins are still localized within a particular cell type, whereas these small molecules can be released into blood, where, again, we can non-invasively assay them.
All right, another question. How many of the molecules that are captured by your mass spec systems are known versus unknown molecules? When you find a novel molecule, how do you go about identifying it?
Sure, and this I really recognize this is an area we glossed over for the interest of time, but I'm happy to chat about this further. So from a typical biological specimen, whether it be a blood, meaning a plasma or serum sample, we can capture somewhere in the order of 11 to 15,000 small molecule biomarkers. Now, as mentioned, among those, say, 15,000 markers, somewhere in the order of 1,000 of them we can readily identify up front using our 4D chemical characterization. That means that there's over 14,000 unknown markers that are typically found in a particular biological sample. As you saw from the examples and the case reports I showed you at the end, oftentimes the most interesting markers are the ones that are most unknown. And this is a reoccurring lesson that we learn in human biology, that the unknown is far greater than the known. And certainly that's the case in this area of biomarker discovery. Now, for any of these unknown molecules, we can use that 4D chemical classification to isolate out the marker and solve its underlying structure using both NMR as well as cryo-EM. We go about the process of synthesizing these markers to confirm them and ultimately use this information for development of targeted diagnostic assays.
All right, another question. You mentioned your biomarker analytical capabilities can detect broad chemistries, but can you give more detail about which particular chemical classes are detected?
Absolutely. So, in many ways, the chemistries that we capture are relatively agnostic, meaning that we can capture very, very different chemistries from very polar molecules such as organic acids and amino acids, all the way up through polar lipids, small oxysterols, eicosanoid, fatty acid derived molecules, bile acids, all the way up through very, very neutral structural lipids, including triglycerides. Now, within there, you can imagine among the chemical classes, we capture molecules from all 14 superfamilies of chemistry, as well as thousands of unknown molecules that fall within different regions. Again, from very polar water soluble molecules all the way through very non-polar molecules through our isolation procedure and ultimately these novel RLC systems.
All right, another question. Can you speak more on how small molecule biomarkers can be used to identify druggable targets?
Sure. Sure. So through these studies, what we oftentimes find is that there's, as you can imagine from the examples we showed you, there's a host of markers that are associated with particular disease processes. Now, in beginning to network these markers together, we can oftentimes identify a singular biochemical or biological pathway that's altered, as is the case with the NASH molecule, for instance, both through classification of these markers as well as through genomic integration. Now, that oftentimes reveals the key target that may be developed as part of a therapeutic program. So in the instance of NASH, we found multiple markers that read out a particular biochemical pathway. Through genome-wide association, we're able to identify the actual enzymatic processes that give rise to that marker. Through Mendelian randomization, we can begin to infer causal relationships between specific markers and disease processes. And then those enzymes and transport systems that were identified through our GWAS systems can then be sort of advanced as part of a therapeutic targeting approach.
All right, more questions here. The technologies capture an enormous amount of data. Can you talk through how you go from tens of thousands of biomarkers to single biomarkers to advance to clinical practice?
Absolutely. It's a great question. And as you can imagine, there's many technologies that generate data. And the goal is not fundamentally to generate massive amounts of data, but to generate singular actionable insights that allow a program to move forward in a very deliberate manner. And the divide between data and knowledge is really really bridged by biocomputational integration. And this is where we spend quite a bit of time at Sapient, not only in generating data, but also in analyzing the data statistically, as you mentioned, Andrea, in order to be able to identify those one or two singular markers that provide actionable insight and allow us to advance forward sort of drug development. Now, the way we approach this is through a variety of statistical approaches, everything from very simple regression statistics to much more advanced patterning and machine learning and AI-based approaches. Independent of the underlying statistics, having a human biological database that encompasses over 100,000 individuals that allow us to cross-validate provides an enormous amount of confidence in any underlying discovery, as well as allows us to advance the biology around any specific biomarker. And this is a way in which we can go about using a single experiment to make an underlying observation, statistically isolate out that singular marker, and then cross-validate it. And that entirety of approach is really represented by this biocomputational integration that allow us to go from data to real actionable knowledge.
All right, another question. How many biomarkers or combination thereof are needed to make an accurate prediction or readout of a disease state, and how many time points?
Yeah, it's a really good question, and the simple answer is that it very much depends upon the specific disease or the specific question. In almost all instances, it's our goal to be able to derive a single marker, not a pattern of markers, but a single marker that we can advance as part of our translational approach here. And the reason this is so critical is that whereas we can measure many thousands of markers, clinical practice really necessitates only a single marker or two markers be assayed as part of a clinical program. And so it's always our goal to be able to get down to that one or two markers that are key in order to translate that discovery. Now, there's certain diseases whereby there may be three or four that each read out different pathways and very complex common diseases such as diabetes, which really represents a number of different pathways and different ways in which an individual can go from a non-diabetic to a diabetic state. There may be four or five biomarkers. For drug response, typically we can get this down to a single biomarker that really reads out responder versus non-responder status for most human diseases. Now, with regards to time point, once again, it very much depends upon the specific disease state and the pathobiology that underlies that disease. And there's certain diseases for which we can identify markers that are altered years or even decades in advance, is the example of NASH. And there's other markers that we measure at single time points after a drug therapy to obtain a pharmacodynamic marker. Again, these are oftentimes dependent upon the underlying therapeutic and the biological process at play. Our goal is always to try to simplify the complex in order to be able to identify a single time point and single marker.
All right, another question. Is your biological database representative of the general population, or is it enriched for certain disease types?
Absolutely. And so across our hundred thousands of individuals that are part of our human biological database, I would say over two-thirds of them represent the general population, in which these are healthy adults from around the world, so genetically diverse, ethnically diverse, geographically diverse individuals who are followed for up to 20 to 30 years for the development of disease states. And that longitudinal information really represents sort of the general population. The other one-third of our database is really made up of individuals that have been isolated out because they have specific pathologies, whether it be rare diseases, as we showed you for the rare mitochondrial diseases, rare oncologic diseases, other tumors, rare lung diseases, neurodegenerative diseases, et cetera, et cetera. And so these are much more disease specific, if you will, around specific therapeutic areas and disease areas.
So we have time for one more question. We had a lot of great questions today and couldn't get to them all, but we will try our best to get back to everyone who submitted personally after the webinar. So for this last question, how do your discoveries complement proteomics and genomics data? Many people are interested in doing multi-omic integration. How do you address this?
Absolutely, Andrea, and thank you again for all the questions today. What I'd say is we spend quite a bit of time working on integrative means that allow us to integrate these small molecules with underlying genomic factors, as we showed you in the example of NASH or the PD-1 target engagement. We can provide similar approaches through the integration of these small molecule biomarkers with microbiome and proteomic measures in the same way, and trying to come up with risk classification scores that leverage multitude of different information sources, whether they come from proteins, genomics, microbiome, and or these small molecules, to both understand a disease process as well as to derive diagnostic sort of metrics. And what I would say is that I understand there's been a number of questions. We're happy to chat with anyone who may be interested in hearing more about these approaches. I would encourage anyone to reach out to us. As shown here on this final slide is our contact information, both our phone number as well as our email. We invite anyone using the QR code here just to schedule a time to chat with us, and we'd always be able to answer any further questions.
All right, well, thank you for attending this Fierce Biotech webinar and submitting so many great questions. I'd like to thank our speaker for participating and Sapient for presenting today's webinar. This webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining, and we look forward to seeing you at future events.
Thank you much, Andrea.